on that? Cool. Go tell the other one. Thank you for that, by the way. All right, let's take a look. Thank you. All right, let's take a look. We're going to get rock and rolling here. So we want to talk about genetics. That's this whole second unit. And genetics is the study of your genes. It's the study of that code. It's the study of that information that makes you who you are. So let's talk about it. And I want to start with just this here, Intro to Genetic Processes. That's a fancy sounding lesson title. Let's talk about it. So before I guess we get to that, we need to talk about this idea of cell theory. And cell theory is sort of what we call the central dogma of biology, which is to say it is one of the two things that sort of from which we gather all the rest of our theories. Does that make sense? The other being theory of evolution is one of the central dogmas of chemistry. So cell theory states what? Well, what do we know? We know that part of our definition of living things is that all living things, yep, yeah, are made of cells. Good. Hello. Hi. I think my watch repair kit's at home. All right, so all living things are made of cells. Sorry, what was that? I don't see that. You should probably look a little harder. So all living things are made of cells. Now what else can we say? Well, where do those cells come from? Every cell comes from where? Guys, do we have to talk about the bird's cells and the bee's cells? Where do all the cells come from? In what sense? Good. Very good. Exactly right. That's a perfect answer. Every cell is the product of a previous generation of cells reproducing. So, keeping in mind that the DNA or potentially the RNA of the cell is the material that contains the blueprint cell, the reproduction of that cell must include the reproduction of the DNA or the RNA. All right, so in other words, the DNA or the RNA contains all the information to run a cell. Right? So as they make a new generation, that new generation of cells has to have a copy of all that information. Which means, of course, before a cell can reproduce, what must it do? It needs to make a copy. If it's DNA or it's RNA. This, by the way, is the one thing that has always fascinated me most. In everything we think about in sort of the theory of evolution and the idea that cells came into being somehow sort of organically through chance, the fact that this ability developed, the ability to make a perfect copy of its own DNA, is just so fascinating to me because it seems like such an enormously complicated process. Though as we learn about the structure of DNA and how it's made, you'll see a bit about how that isn't as impossible as it might seem at first. All right, so we want to kind of differentiate between two different sort of methods of reproduction, and the first we want to look at is asexual reproduction. What's that mean? Yeah. I am so sorry, I couldn't hear you. Okay, so a single organism required. In other words, it doesn't require a mate, doesn't require a partner. True? What else? Yeah, I'm going to say results in daughter cells that are clones of the parent. What does clones mean? Yeah, specifically identical copies. Identical copies of the parent cell. There's a company now in China, you can send them some DNA from your dog and they will clone your dog. It's like when Arlo passes away, if I had $230,000 to burn, I could be like, make me another Arlo. I don't have $230,000 to burn, but you could do that. Barbra Streisand had her dog cloned. Singer, Barbra Streisand. Famous entertainer. Anyway. She also has a mall in her basement because she's like too famous to go shopping, but she missed shopping. So isn't that kind of cool? A neat idea. So like all her stuff, like, so it's not like a real mall, obviously, but like basically, so like all like, she has like a museum of like all the costumes she wore in like her stage plays and like her movies and stuff. But instead of just putting them on display, she had it made as if it was like a clothing store, like a shop with all of her costumes there. And then like all of her books, instead of just having them on a bookshelf, she had it made like a bookstore and so on and so forth. So like all of her stuff is displayed so that like she can like quote unquote go to the mall. Sorry? I don't know. You'd have to ask her. Call her up. Be like, Barbara, talk to me. Sorry? I know you should. Are you interested? I mean, I'd be interested. Sign me up. Live in California. Be the cashier at a mall with only one customer. I think I could handle that. Anyway, maybe I couldn't. I don't know. Never worked retail. Sorry? I think I could do it. And then I'd say here, any changes from generation to generation here in asexual reproduction are the result of mutations. And we'll talk more about this later, so I'm not going to define it too much here. But really quickly, what's a mutation? What's that word mean? My sister. Wow. Anyway. Yeah, a change in the DNA that happens by random chance. It could happen by UV radiation damaging the DNA, or just a mistake during replication, or a bacterial infection or a viral infection of some sort. So yeah. All right. And it happens by the process in eukaryotes. Asexual reproduction happens via mitosis. Okay. What can you tell me about mitosis before we go to the next page and before we write it all down? Who can run it down for me? What's mitosis? Talk to me. Sure. So one cell makes two daughter cells. Good. I like that. What else? It has organelles, so they divide it. They make copies. Yep. Absolutely. What else? Yeah, what's that mean? Because that's true. Cell spends most of its time in interphase. Yeah, interphase is just what we call when it's not reproducing, which is it's interesting actually. It tells you really something. Like imagine like if every minute of your life you weren't busy actively delivering a baby, they're like, it's just like the interphase. It's just like the part of your life that doesn't matter. Kind of reductive, right? Like that'd be an insulting thing to say about a human. So I've never really liked the name interphase. I think it's kind of silly. Because, of course, there's plenty of cells that spend, vast amounts of time not reproducing, right? And so just saying they're in interphase, they're just waiting to reproduce, has always seemed silly to me. But yeah, that's true. We call them living the normal cell lives just interphase. Yeah. We're going to talk a lot about mitosis, and as soon as you understand what the steps of mitosis are.
Sorry for the interruption. The following students need to head to the gym: Olivia Bicker, Deanna Shikaki, Andrea Lichard, Devago Flowers, Ozzy Grenier, and Savannah Mann. Thank you.
No, it just keeps happening. I get distracted, and then I lose track of where I was thinking. Anyway, so yeah, I think once we get mitosis really clearly in your head where you understand the steps, then you'll know the difference, because you're going to find that the difference is just one is what eukaryotes do and one is what prokaryotes do. And if you know the steps of mitosis well, you'll be like, oh yeah, well that is all about how the organelles are reacting to this stuff. And since I know that prokaryotes don't have organelles, they don't go through mitosis. Does that make sense? I think just trust the process. Trust that by the time learning it, you'll know the difference really well. In other words, put a little bit of faith in my ability to teach you.
Oh yeah, you look like Superman.
If you were to say that a few seconds ago, I would, but you kind of just
Hi.
Bye, Mr. Orlando.
Why is he walking like Goofy from Mickey Mouse?
Well, boy, am I going to have to go look at my Bria sign? Remind me. Anyway, enough about my crushing realization that my life's work is for naught. Let's get back to mitosis, shall we? It's the mad cow. So listen, most eukaryotic cells undergo a process called mitosis. Sure, yeah, you do what you gotta do. This should be a review from grade 10, but long story short, it's just asexual reproduction for eukaryotes. And the reason we say only some cells do it is because not every eukaryotic cell reproduces. And some reproduce, but reproduce sexually. So that is relatively few, certainly in our body. Okay. Right? So the first thing to understand is that during normal cell life, which is just called what again? Yeah, during interphase, the DNA is in loosely coiled, in a loosely coiled form called chromatin. It's important that the DNA be kind of coiled because in every one of your cells, you have about a meter of DNA. Somewhere between a meter and 2 meters of DNA in a human cell. And the problem with that, of course, is how big is a human cell? A lot smaller than a meter. And so how do you fit something that's a meter long inside of something that's very tiny? No, shrinking it is not an option, but you can compress it. And how do we compress it? By coiling it, right? You guys don't remember telephones that have cords because you're not old, but I do. And so there you go. So you've got your little telephone receiver here, and then you've got the little box here, and it's got the buttons on it, right? And how are they connected? They're connected with a coil, but you wanted the coil to be long enough that you could step away from the kitchen and talk to your friends in the living room, but you don't want to have a big 20-foot cable dangling from the thing all the time, so it's coiled like that, and then you can stretch it out, and it can go a long distance. So you walk back, it coils back up.
A single cell of DNA is really small, but in the whole meter DNA thing, that's just a really abstract construct.
Yeah, it's a really weird. So basically, you've got a cell that's this big and a chain of DNA that's that long. How do you get that into that? And the answer is what you do is you kind of coil it up.
Yell at it.
And in fact, you take that coil and you make a coil of that. So that's coiled. And this is what we call supercoiled. Clever name, right? But even supercoiled, it is still a fairly loosely packed thing. And that DNA is found, if this is the cell, what do we call this structure inside the cell? The nucleus. And that DNA just exists as a big, long chain here.
It's so hard to
So we have 46 unique chains. I shouldn't say unique, actually. We have 46 chains of DNA loosely coiled. That's for humans. Different eukaryotic species have different numbers of chains of DNA, but in the nucleus. The coiling fits 1 meter of DNA into a tiny nucleus. But it's loose enough that the DNA can be accessed to be read. Because remember, this is constantly in use. The cell is constantly coming in and saying, oh, hey, we need this gene right here. So we're going to go in and we're going to read it. Does that make sense? And you have machines in your cell whose job is to go read that DNA and then carry out the instructions on it, which we'll talk later on about what the instructions on DNA really are for. But that's the idea there. Does that make sense? So coiled and supercoiled enough that it fits in the nucleus, loosely packed enough that you can still get at any individual segment of the DNA and read it and use it. But listen, who here has ever moved? I've moved 6 times in my life. Yeah. Yeah, so if you're moving, what do you do with all your stuff? You pack it up. Why do you pack it up? Yeah, that's right. It's really hard. If you tried to move without packing your stuff up, like you're going to run out to the van with like a shirt and then like go back in and get like 2 more shirts, then 2 more. You know what I mean? Like you got to pack stuff up so you can get it transported. And of course, also if you have valuables, if you have stuff that's breakable, if you have stuff that, you know, you need kept together. And when you get to the new place, you're going to probably take all your stuff and move it all to your room. And like your siblings probably going to take all their stuff and move it to their room, and you're probably going to take all the stuff from the kitchen and move it to the kitchen. So if you pack stuff up, it helps that process, right? Does that make sense? So, likewise, at the end of interphase, in preparation for replication, the cell does 2 things. And the first is pack the DNA way more tightly into chromosomes. And so you can see under a microscope when a cell is about to undergo mitosis, because you can see that the nucleus all of a sudden will contain distinct chromosomes. And so what you'll see is instead of just this loosely packed here, you'll actually see these individual lines of DNA that look like that. And I'm only going to draw 4 of them just because I don't want to draw, but actually in a human cell, we would have 46 chromosomes. But the second thing that happens is what? The DNA is replicated, which means what? Duplicated, I should say. What's it mean to duplicate something? To make a copy of it. And so what happens actually is that inside the cell, inside the nucleus, we have these chromosomes, and now they actually appear like a little letter X. They are stuck to a perfect copy of that chromosome. All right? So each chromosome, yes. I'll get you one in just a moment. Oh, I was just going to grant you one, but we're good there. That's good. So in this case, the DNA is now too tightly packed to be read. Does that make sense? So the DNA can't always be packed up like this because then it can't be used. Right? It can't be used to read and monitor the cell. So this just happens right at the end of the cell's life cycle when it's ready to reproduce. It's like packing things up. It's like in your house. You pack everything into a box. Can you use all that stuff? No, but it's good for moving, right? That's like this. We pack up to form the chromosomes, and the DNA can't be used. So now, let's talk life cycle. And this is not necessarily an accurate proportion, by the way. But so listen, during the G1 phase, this is normal cell life. Before the DNA is duplicated. The S phase stands for synthesis. And this is where the DNA actually gets duplicated. Synthesis because there's more DNA made. So this is where the duplication of the DNA occurs. And then here we actually get G2, which is basically a growth spurt in preparation for division. And I'm going to say in preparation of mitosis. So don't worry, I'm going to write this and give you a minute to write it. But this is normal cell life. And I'm just going to say, see the stages above. G1, S, and G2. That's the worst looking G I've ever seen. The specific names aren't the important part. The idea that's the important part is to understand. I don't want you to treat understanding mitosis like memorizing a bunch of names, because that's not the point. The point is to understand that the cell is undergoing this complicated process. And really the point is to understand, why is all this necessary, right? Why doesn't the cell just kind of grow and split in half? Well, it does, but when we start to break it down into stages, we realize that there's a lot of work that has to go into doing that, right? You can't just split in half, because if you just split in half without dealing with the DNA, one of the cells would have DNA and the other one wouldn't. Well, that's not going to give you generations of evolution, right? That's just going to not work. Yeah. No. Where was I? No, that's okay. I'll distract now. Sorry, and then, okay, if we're going to make a copy of the DNA, well, even still, what's the DNA held in? The DNA is held in a nucleus. So you still can't just split the cell in half, because now you'd have one cell that had a nucleus with 2 copies of the DNA, another cell that still has nothing. Does that make sense? So how do we deal with that? So that's what understanding mitosis is about. It's not really that the steps of mitosis are that important. Rather, it's one of the first...
The following students, please need to go to the gym. Jason Spry, Natalie Stratichuk, Haley Tupper, Jaden Van Kloop, and Alex Rioscos. Thank you.
Oh, okay. Where was I? Oh, yeah. Oh, is that where I was? I don't know where you were, Lana, but I know where I'd like you to be, which is moving. Okay, so listen. Shh. The important thing about mitosis is it's one of the first times we start to learn about the way the cell actually does something. And the thing you realize is the way cells do things is complicated, right? There's a lot going on. Does that make sense? That's the main thing I want you to take away from this. There's a lot going on. So during normal life, the cell begins to slowly prepare for reproduction. So it's not like mitosis happens like, boom, here we go, and the DNA replicates. The truth is that during its life cycle, it begins to make preparations for these things. That monitors what happens on a big scale with sexual reproduction too, right? Like, it's not like human beings one day just, boom, wake up and everything that's involved in sexual reproduction happens, right? Like, there's a slow, gradual process that leads to those systems functioning properly. Yeah. So now, mitosis itself is the active stage where a cell begins to actively divide. So let's take a look at that. You know the stages. So now let's take a look at them in a bit more detail. So the first stage is prophase. And in prophase, 3 major things happen. And all of which make perfect sense if you think about what has to happen. If you think about mitosis in general, the job is to start with 1 cell and end up with 2. When we leave interphase, we already have duplicated the DNA. Does that make sense? That has happened. So now, all we have to do is get 1 copy of the DNA to one side of the cell, 1 copy of the DNA to the other side of the cell, and then split them in half. Does that make sense? That's it. That's the whole job. So to make that happen, what's going to happen? Okay, but before they can go on each side, what's going to happen? Already happened. That actually doesn't happen during mitosis. Like we said, that's one of those things that happens gradually. During interphase, the DNA duplicates. So that happens during the S phase of interphase. In other words, we said like that can happen gradually during the lifespan. But now when it's time to cut in half, what's going to have to happen before 1 copy of the DNA can go over here and the other copy can go over there? Kind of sounds to me like you're reading off my note. But don't read off my note. Just think it through. You're an engineer, and you've been given the task of taking a cell and cutting it in half. Right? You want to move some of the DNA over here, some of the DNA over there. Cool. I go to move the DNA. So I reach in with my little tweezers, and I go to move the DNA. What's the problem? Very good. It's an organelle called the nucleus. So if we're going to take the DNA and move some of it over here and some of it over there, we've got 2 choices, right? Really, which is we could split the nucleus in half, right? We could divide the DNA inside the nucleus and split it. Or we could deal with that later and just get rid of the nucleus. Which one does the cell do? Gets rid of the nucleus. So that's the first step. In the first step, we start off with a cell that looks pretty much like a regular cell, except the one thing we want to note is that this chromatin is actually stuck to a copy of itself. And they're not really obviously different colors. But I'm just showing that by kind of showing the original as pink and the copy as green. So as prophase begins, 2 things are going to happen at once. The nucleus itself is going to start to degrade. It's going to start to break down. And while the nucleus degrades, the chromosomes are going to form, which is the lengths of DNA condensing into this much more tightly coiled structure. Right? And at the same time, what else are we going to want to do? Well, we said we want to take them and we want to move them. How do you move DNA? How do you move anything? With your hands? The cell doesn't have hands. What does the cell have instead of hands? Okay, is it going to like reach its flagellum inside and like start grabbing stuff? Let me tell you, that would be cool. That would be cool. Yeah. Okay, but that's just liquid. Is it just going to wait for them to float? It's just going to be like, float. Yeah. No. But actually, flagellum's a really good answer. Does anyone remember what flagella are made of? We mentioned it like super briefly, like just like it was in a slide in one of the pictures, and I mentioned it. But it's made of something called microtubules. It's made of microfilaments, which are basically just these like little small protein chains that cells make that they can move. And so they make sense for flagella or cilia because they can move things. Well, that's actually basically what the cell's going to make. So it's going to start to form these structures on the poles of the cell called centrioles. All right, so that's called a centriole. So the centrioles start to form, and the nucleus starts to dissolve, to degrade, and then by the time we're done prophase, what has happened is the nucleus is gone, and the chromosomes are now kind of freely floating about in the middle of the cell, more or less. And again, they're not actually two-tone like this, but I'm just trying to show that things are in pieces. And we have the centrioles, and they begin to make these little fibers. Basically, they're going to make little like protein ropes that they can use to grab onto stuff. And those are going to start to grow out. These are called spindle fibers. That's called a centriole. And that is called a chromosome. That is the worst writing anyone's ever done. Let's try again. I want to look, by the way, really quickly at chromosome, just what it looks like. The chromosome is basically this X pattern of DNA that is like really, really, really tightly packed. And then they're held together with a structure in the middle. And the structure they're held together by is going to be a good example of something that happens a lot in science called names that are horribly sound alike to other names. So the things that make the spindle fibers are called centrioles. That makes perfect sense because they're on both far ends of the cell. What I'm saying is, as a name, I don't think that makes sense. Awatrioles would be better. Far end trioles. But no, they're called centrioles because they're not in the center. Annoying. But what do we call this middle piece that holds the two copies of the chromosome together? The centromere. Good. And the centromere holds the duplicate copies of the chromosome together. You could almost think of it like if you took a piece of paper and photocopied it on the copier, if the copier automatically stapled your original to its copy, that would be like the centromere. It's basically a staple that holds the two copies of the DNA together. Does that make sense? So as the DNA gets duplicated, it's held together in this central spot by the centromere. So by the end of prophase, what we have is the chromosomes are no longer in the nucleus. The spindle fibers have begun to grow from the centrioles. And again, don't memorize this as like, oh, there's centrioles and centromeres. That's not the way to think it. And I think this answers your question from before. Rather, understand this happens because of course it has to happen. This is an engineering solution. Does that make sense? The cell has a problem. It needs to make a copy of itself, which means it needs to make a copy of its DNA, and it needs to split in half, and it needs to make sure that both halves end up with a copy of the DNA, right? That's what reproduction is going to look like. And if you're going to get new cells, the only way you can do that is by taking the old cell and cutting it in half, but making sure both of the new cells have a copy of the information that says how to run the cell, right? Is that logical? If that's logical, then you don't have to memorize this because it has to happen. You just talk yourself through it. Well, if both cells have to have a copy of the DNA, then the DNA has to be duplicated, right? Okay, that happened during interphase. Well, if both cells are going to end up with a copy of the DNA, it has to get moved to both sides of the cell so you can split it in half. Okay, oh, it's in a nucleus. How are we going to solve that problem? Well, let's get rid of the nucleus, right? Well, what's going to move them to the right spot? Well, we better build an organelle whose job is that. That's the centriole. The centriole's whole job is to build these little tubes that are going to go out and grab the DNA and pull them to where they need to go. Does that make sense? So don't memorize it as steps. Think of it as an engineering solution. It's a lot easier to understand what all the parts of a car do if you think about what a car's job is. You know what I mean? Like steering wheel, gas pedal, brake pedal, blinkers, windshield wiper button. You don't memorize what those things are. You're not like, and this thing's called the steering wheel. What does that do? Be like, oh God, I got to check my notes. I can't remember. It's in the name, right? Like you know what it is. It's the thing that turns the car. If you think of this not as a bunch of stuff to be memorized, but rather as an explanation of like solving a problem. The cell has a problem. It needs to duplicate. How do we solve that problem? It's so much easier. Does that make sense? So now, what's going to happen next? Don't read the note. Don't look at it. Just in terms of what the cell has to do. What's going to happen next? Yeah.
The fibers are gonna stretch out.
Good, they're going to have to grow out. And what are they growing out towards? The chromosomes, the DNA. And what are they going to do when they get to the chromosomes? Yeah, they're going to like try to pull them away, right? Which means they're going to have to latch on. Does that make sense? Before you can pull something away, you got to latch on. And what part of the chromosome, if you had to guess, is designed to have these spindle fibers latch onto it? That's right, the centromere there. By the way, a bunch of these spindle fibers will grow out and never grab anything. That's fine. When the retraction stage happens and they get pulled back, they just get pulled back attached to nothing. Does that make sense? Because the only thing that they're designed to actually grab onto is the centromere. Does that make sense? So this thing basically just sends a bunch of feelers out and says, hey, let's find centromeres, and when it finds them, it grabs onto them. That's all. So now let's draw the next stage. At metaphase, we start off where we left off, which is we no longer have a nucleus. What we do have are a bunch of chromosomes in the center of the cell, like this, right? Oh, why did I erase that? Because I'm silly. That's why. I'm a silly goose. Aren't I indeed? And these start to grow out, and they're going to attach. And what structure are they attaching at again? Yeah, but I can't stress there's a bunch of these that go out and don't attach to anything, right? And that's okay. The ones that find their way attach. But of course, what's happening here? They are also going out and they are attaching as well, right? Okay, so now that that happens, they're going to start to pull and retract. But now, if these are getting pulled on from both ends, what's going to happen to them? If I took 5 of you and the rest of us split up between each, or 4 of you, and the rest of us split up between each half, and we all threw ropes out and hooked you with them like lassos, and we pulled from this side and they pulled from that side, where are you going to end up? Before? In the middle. Does that make sense? As we pull, they're going to, before they split in half, they're going to end up making almost a perfect straight line down the middle. And so by the end of metaphase, that's what we see, that the chromosomes basically line up perfectly down the middle of the cell because they're being pulled in both directions at once.
Well, I know you don't suddenly have an installed soundbar in here, do you?
In here?
Yes.
No.
Okay. That's what I thought.
Why?
Board wants to know. I'm checking. I have to check every classroom.
For sound bars?
Soundbars and projectors.
Interesting.
We're just doing a baseline head count,
Cool.
so we can figure out the next phase of technology for us.
Cool. Yeah, I have a sound bar.
You have a soundbar?
Sure. Yeah, say that. I bought this with my own cash, so tell them that we have one here that's provided, and then when money comes up, we'll get a replacement for the one that doesn't exist, which I think is fair. Seriously. Listen, if that money doesn't go to Eastdale, it'll go to somewhere else. And you know where I want it to go?
Still.
That's right. Okay, so by the end, we get the chromosomes lined up along the center of the cell, which we refer to as the equator of the cell. Does that make sense? The equator, which makes sense, right? That's what we call the central line of like our planet. So at the end, by the end of metaphase, the chromosomes are lined up at the equator of the cell. But again, don't memorize that. It makes sense. If you latch on to the chromosomes and then start pulling in both directions, yeah, of course, they're going to end up getting basically pulled to the middle, right? Okay. What's going to give here? First of all, why are we attaching on both sides? Don't we want some of them to go this way and some of them to go that way?
Well, they're duplicates, so you want them to split up.
Very good. We want all of them to go both ways, right? Which is to say each chromosome is made up of 2 copies of the same thing. So what's going to happen next? They're going to pull apart. And half of each chromosome is going to go to one pole, and half of each chromosome is going to go to the other pole. And indeed, that's what we see. Can I move on? Do we have this? I don't want to rush you. All right. So then, finally now, we're going to start to get the shortening of those spindle fibers made possible by the fact that the chromosomes are going to actually split. And I want to show that it's not necessarily the case that it's always the same one that goes to the same side. Do you know what I mean? There's nothing, the spindle fibers don't know which copy is the original and which copy is the duplicate. Did I get? Oh, I did get it right. Okay, look at that. Until eventually we get the shortening down, where over here we've got these chromosomes, over there we've got those chromosomes. Just like that. And the spindle fibers will then start to dissolve because they've done their job. Right? Ready to dissolve because you've done your job? That's how I would feel if I'd done my job, but I've been told recently that I make everything more confusing. It's causing me an existential crisis. I need to find myself a cake. I'm going to eat that cake, sadly. Sad cake eating. It's one of my specialties. Oh, I'd like that. Okay, let's focus. What? No, listen. Let me say two things. First, that's why they have menus in restaurant. Different people like different things. Secondly, you're wrong. But firstly, it's important to understand that people like what they like and they don't like what they don't like, and that's okay. So, like, you know. Sorry? Yes. And if I tried to think about it and say it again, I would doubtless fail, but no.
Can you say it again, please?
No.
I didn't hear what you said.
Brady, I'm not your dancing monkey. Thank you. All right, now what's going to happen next?
Are you dancing?
Oh.
I don't dance. You don't want to see me dance, I promise. I looked good. Okay, what's the next step? What's going to happen? And again, don't read it to me. Just think it through. What's going to happen next? Very good. We're going to get what we call the cleavage furrow. Cleavage just means breaking in two, right? So when you break two things in two, a meat cleaver breaks one piece of meat into two pieces of meat. So we're going to have this thing basically pinch in and start to break into two pieces. Because cell membrane, do you know what cell membrane basically is? There's something that you've all used in your life, and it's a delightful, joyous thing. No, I mean, a joyous, delightful thing. Basically a soap bubble. And the reason you think of soap bubbles as being really delicate and easy to pop is because when you make soap bubbles, they're enormous, right? And so they've got a huge surface area that, you know, you can break easily. But basically, a cell membrane is that exact same material, only it has to only form a bubble that's really tiny. And at that scale, its surface tension is very strong, right? When you take that surface tension and spread it out to the size of like a bubble that's like visibly this big, that's now a very delicate thing. But at a scale where it's the size of a cell, that's a very tough thing. So as weird as it is to think, all of your cells are little soap bubbles, kind of. Weird, right? I'm a very big soap bubble. Thank you. Shh, quiet. So now if you take a bubble and you squeeze it in on itself, you know that you can make one bubble into two bubbles. Or conversely, you can do the opposite, right? You can have two bubbles form, and if they form in the right way, they can join together and make one. You ever seen that happen in either direction? Well, that's what's going to happen here. If you just pinch the sides of this in, it will start to form into two separate cells. Now, at the same time, what do we almost immediately want to make sure we're doing with that DNA? Sure. Yep, definitely. That's why we moved it to both poles. But what else do we want to do with that DNA? Very important. Oh, come on. Yes, you do. Protect it. And how do we protect the DNA? By adding membrane. What do we call that membrane? No, cytoplasm is the fluid inside the cell. Yeah, we're starting to form a new nucleus, right? That's very important. I think I put 2 green and 2 pink in each one, right? Then by the time we get here, we should have a nucleus forming on each end, and we should have the cleavage furrow starting to develop, which is going to give us two separate cells. All right, so we get the reformation of the nuclei. I'm going to say nuclear membrane. We get the cleavage furrow dividing the cell. All right, and that's what we call telophase. The spindle fibers have broken down completely. We get the formation of the new nucleus. I guess we could show at the beginning there's still some spindle fibers present, beginning to break down. By the end, they are gone. I'm pretty good. Pretty good in time. I could be great. Oh, well, I wouldn't want her to be intimidated. It's her mental health and sense of self-worth is very important to me, and I would hate for her to be like, oh man, all this time I thought I was pretty good at art, and then this guy shows up out of nowhere, a science teacher, and just blows me out of the water just day one, right? It would hurt her feelings, and I would never want to do that to a coworker. So no, I'm good.
Mr. Landa, when I had Craig, I made this a teacher-able computer.
Yep.
So all I need to add is so that she can print if and
Yep.
is that? I know you've taught me.
No
I
worries.
should
I
be
got
able
it.
to do it.
I got it.
Sorry to interrupt your
You're
class.
fine. Guys, what's the next step? Yeah, cytokinesis, which is what? Yeah, and cytokinesis is just the actual, this is what we call it when it's no longer a single cell. Mitosis actually doesn't include the portion where there are actually two separate cells. So by definition, it's kind of a finicky little definition, but it is important. As soon as there are two cells, we are in what we call cytokinesis, which is just them moving apart from each other. As soon as the cells are separate, A-R-A-T-E or E-R-A-T-E?
Separate.
A-R-A-T-E. Yeah. They are in the stage of cytokinesis. And they just move away from each other. But again, I can't stress enough, don't memorize it. Don't think of it that way, but rather talk your way through it. Oh, this has copier 1 and copier 2 on
Oh
it.
shoot! So I went to print something
That's
and
weird.
it wouldn't let
So
it.
let's see.
So yeah, I went to print that, and then it would only give me when I went down see more.
Oh, that's weird. Okay, well let's. I'm just gonna remove it and re-add it.
Oh, good. Thank you for asking. I've been on the treadmill getting my joint moving, and it's doing much better. Thank you for asking. How are you? How's the head? Again.
I haven't seen it actually.
Oh, okay. Okay.
It's weird that that's very strange.
Let's maybe. Is it okay if I keep her for a few minutes into fourth and then send her down
Yeah,
to you?
of
Okay,
course.
just while I have her there.
Seems good, eh?
Oh, so good.
Yeah, already
Yeah, jump right in with the kids. Like,
already feeling confident.
yeah. When she addressed the class, nice, because I had her introduce herself, and she had a good projection of her voice, and all the things.
Yeah, no, she's great. Yes.
Is this for you, bio?
3U.
3U bio?
Yeah.
You got a nice class.
Really good.
I was like, okay. He's counting to 10 and ran whatever he ran in 5 seconds. He's like, see? And then faster than 10 seconds. Oh, that's so cute.
Did you introduce her to all the bosses and stuff? I think she said she meant thank you for doing that.
Why can't you connect the printer?
Should I bring this to you later?
Yeah, catch me later.
Yep.
We'll catch it.
Yep.
All right.
I'll have her bring it to you when she comes.
Oh, that'd be great. That'd be perfect. Okay, so guys, listen up. This needs to be prior knowledge for you. What do I mean by prior knowledge? Yeah, it means that going forward from here, we're going to talk about a lot of processes, and I need you to have a good, solid grasp of this. And again, that doesn't mean I've never found any value in being like, oh, it's PMAT, those are the stages, and trying to memorize it as a picture. In biology, what we're learning about is processes. Does that make sense? How do things work? It's an engineering problem. What is the goal? What is the cell trying to do? What are the steps it follows to do it? And if you forget the name of something, I'll remind you of the name of it. Whatever, that's fine. That's easy to work with, right? But what you need to understand is the principle of it. What is happening? Why is it happening? And if you understand that, then who cares if you mix up your telophase from your anaphase? Who cares? I mean, I'll still mark you wrong, but I don't care, right? And you shouldn't care either, because the important thing is to understand the idea. And the idea is that this is a very complicated system. Even here, by the way, I hope you see that, like, every single thing we describe, like, you see this here where we're like, oh, hey, these things form, and then they start growing little tubes that go out and reach out and grab a chromosome. That could be your life's work, right? There could be somebody who their 60 years as a scientist is spent studying how the heck does that happen? Do you know what I mean? This stuff is complicated, and it's a big, messy system. And as a complicated, big, messy system, what I really want you to take away from that is that the cell has these really intricate processes, because anything the cell wants to do, because it is a big, complicated machine, it has to deal with it being a big, complicated machine. So if you want to make a perfect copy of a big, complicated machine, there's a lot of crap you have to deal with. Does that make sense? So even this, as the stages of mitosis, just scratches the surface of how this whole thing works. Like, we just hand wave, oh, by the way, the DNA makes a perfect copy of itself. How's that happen? Don't worry about it. You can spend your whole life studying that. Do you know what I mean? Immensely complicated. Little things form, and they form tubes that seek out chromosomes and pull on them. Oh, okay. Yeah, that's cool. Oh, the chromosomes split in half, and one half goes this way and one half goes that way. How? Right? Like, all that stuff is so complicated. So I hope you see that anything you study in biology, every question you answer just gives you 25 more questions. Does that make sense? And that's okay, because your cells have had 4 billion years to get complicated. Put that away. And having 4 billion years to get complicated, it takes a long time to understand just how things work. And in everything to do with cell biology, we have a pretty good grasp of how things work, and we probably know 1% of 1% of how things work. Does that make sense? Like, there's just so much nuance and detail. And of course, cells are very hard to study because they're very small, and we're very big, we're very large. So we're going to do a few labs during the course of this year that show you a little bit of what goes on with studying things, but you're going to find studying how an animal works on the big scale is pretty easy. But then when you try to take the next step and say, okay, what's happening inside the cells of this thing? Well, that's a whole lot harder, right? We're going to do dissections, and we're going to look at, like, gross anatomy and physiology. And by gross, I don't mean like I mean like big scale, macroscopic physiology and anatomy. But when you go to look at cellular anatomy and cellular physiology, what's happening inside the cell, you've looked in microscopes. Like, when you looked at the onion cell and the cheek cell, what could you really see inside? Could you see the Golgi apparatus and the endoplasmic reticulum and the rough ER and the smooth ER and the vacuoles and the lysosomes and the spliceosomes and the individual chromosomes? Of course you couldn't. You saw, like, little brick-shaped things that had, like, a little dot inside of it, right? So you can imagine how sophisticated it has to be. And those, by the way, are really big cells. It's hard work looking at cell biology. Really complicated stuff, but really interesting, really worthwhile stuff. So when you go to study this, my advice would be draw it out. Draw it out from memory. And as you're drawing it out, name everything, explain what's going on, but more importantly, explain what's happening and why it's happening. Okay, the nucleus dissolves. Why does the nucleus have to dissolve? Well, because you can't move the chromosomes if they're inside a nucleus, right? Well, then that makes sense. The spindle fibers start to form. Well, why do they start to form? Well, something's got to move the chromosomes. They're going to be the things to do it. Does that make sense? Just work with it as a process. All right, I'm done. Nothing more today. I'm going to give you guys your tests back. They were very well done, overwhelmingly, very well done. The multiple choice, I didn't mark which were right, which were wrong. We'll take them up tomorrow quickly. I just gave you a grade as a percentage on that. Throughout the test, take it through and give it a reading. You might disagree with me. Minds can disagree. For a couple of the questions, Like, for example, the not the dichotomous key, the one where you had to put in the different traits. There were a couple different versions of that where I thought that's a good, reasonable answer and I gave full marks to. Other places I took off. I used my judgment. Your judgment might disagree. Feel free to come talk to me. Don't be like a Mark Grubber. You don't want to be that guy. But if you have an honest concern that I wasn't fair in your answer, you don't understand why I gave you the grade you gave, come and ask it, by all means. Don't be shy. And when in doubt, come ask me. I'm always happy to explain. Likewise, using the dichotomous key to tell the difference between the flowers. Well, it could be sneaky. Like, there is a right answer. That's a real dichotomous key that comes from a real, like, Ontario flower guide. But it's not the easiest thing in the world to read, right? So I tried to be generous and considerate in how I marked things. Also, I will guarantee, like, I had a sick kid, like, on my lap, like, being a huge pain in the butt while I was marking these. So I may have also just made a good old-fashioned mistake where I just was like, oh, that's wrong, but really it was right. So by all means, you should check your work against my answers. Like I said, don't stress about the multiple choice. We're going to take those up tomorrow. But yeah, so Aruba, come on up. And Charlotte. And Sarah.
Sorry for the interruption. The following students must come to the
Sarah. Mary.
Dawson,
Ruby.
Marshfelder,
Sierra.
Ines,
Hey, Brady, nice work.
Justin, Daniel,
Ruby Elliott, good job.
Spry, and Jaden.
Thank you.
And
Thank you.
Peppy.
Sierra.
Thank you.
There you go, kiddo. Manat is not here, right? Gage. Bria. Bria? No, Bria. Deandra. Which, by the way, Deandra, if I can have that picture, I would love to have it. If you want to disconnect it and give it to me, I loved it. It's beautiful. It's wonderful. Natalie. Mubin. Damien. Samantha. There you go, Mubin. There you go, Damien. You're not Damien. Samantha. Natalie? Oh, right. Alexander, also called down, right? Isaiah. Lisa. Cora. Lila. There's that. You're welcome. There you go, Lila. There you go, Lisa. There you go, Cora. Thank you. It's really, I'm so impressed. Yeah, but like not, again, it's not how I feel at all, but it was really funny. Made my day. Is there anyone I didn't get it back to who, I mean, other obviously than people who just wrote, which I'll get yours marked as quickly as I can. You all done that? Thank you. How was that? Yeah, it felt good? Nice. Glad to hear it. I got those marks put in. I'm just gonna double check the last bit of it. Had I set tomorrow as the due date for the physiology project? Or not the physiology, the pathology project, like the 3 diseases project, rather. Okay, so make sure that's into me tomorrow. Now listen, look at me. We're getting into, I'm going to start treating you like big boy and big girl students, all right? So, unless you've made some special arrangement with me, and by the way, if you got things going on in your life, make arrangements with me. I'm not an unsympathetic person, but outside of that, tomorrow I'm taking that whatever it looks like. And if you don't give it to me, it's 5% a day. But not only that, it's 5% a day, but once I've returned things, you're done, right? Chance gone. And this semester so far, I've been pretty good. I've been pretty fast on my marking. Have I been next day on everything? I think so far. And I plan to keep that up. Because again, I can't stress enough 3 things. First thing, you do yourself a grave disservice if you let yourself start taking extra time on things. Because before you know it, instead of having 3 assignments due or 4 assignments due, you have 8. And then before you know it, 8 turns to 12. And you know what? It's impossible to get caught up once you've got 16 things overdue, right? Like, you just can't do it. It's too much. It's too stressful. But the second thing is, I can't in good conscience start returning things to people when I haven't collected yours. Because then you can see what the grading, then you can look and go, ooh, yeah, got to include that, got to include this. And all of a sudden, by being the person who didn't have things done, you have an advantage over everyone else. So that's not fair. That's not right. Does that make sense? So again, you got something going on, that's a different story. Come talk to me, and that doesn't mean you have to spill your guts and tell me everything going on in your life. I respect people's privacy. But failing that, when it's due, I'm taking it. Period. Got that? Excellent. Which also means if you need help with it, come get help with it. I'm here for you. Yes, ma'am.
What is this? What does this mean?
Oh, well, these should all line up at the same spot because this represents like time, right? So like even though this is human, like kangaroos didn't like go extinct anciently, like they still exist. And like snapping turtles still exist. So they should all just be kind of lined up in line with each other.
Okay.
But other than that, your setup was good. And then was there anything else in there? Yeah, that's all.
Also, what is I'm confused with the marking just because like it's.
Oh, guys, sorry. If I haven't explained this to you, I should mention it now. I mark every question out of 10. And then how many marks you get for it is just, if this is like, if you got 8.5 out of 10 on a question worth 5, then like in my computer, it just multiplies that by 8.5 divides by 10, and that's how many grades you get for it. And I just do that so it gives you a bit more nuance. Because I always find a question that's out of 2, it's always like, well, if it wasn't a great answer, is my option 1 out of 2? Well, a lot of not great answers are a lot better than 50%. You know what I mean? So this just gives me a bit more freedom to kind of always be like, yeah, this is where you're sitting at. Does that make sense?
Yeah.
Cool.
Also, like I'm gonna turn it into but if it's like, does it have to be turned in before 12, or if I turn it in tomorrow night? Like
I'm okay with that. I'm okay with that. Yeah, I'm fine with that. Yeah, did that answer your question?
Yeah.
And like the out of 10 thing is meant to be like a benefit to you guys, not a hurt. What I found is that I find that I give slightly higher grades, but I tend to be giving more of the grade I mean to be giving. Yeah.
Which one's my actual grade?
That one. That's what you got on the multiple choice. That's your actual overall grade.
Thing at the end of 902.
Sure. Yeah, that's fine.
On Mary's, it says that the prim is correct.
Okay, so did I get it wrong? Sometimes I get it wrong. Well, let's follow it. Let's do it. So, narrow leaf, bell-like flower is the bluebell, definitely. Trumpet-like flower, the wild daffodil. So that should be the wild daffodil, right? Okay. So now, broadleaf, heart-shaped leaf is the lesser blah, blah, blah. So that should be the lesser blah, 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 blah. Club-shaped leaf should be the primrose, and the one where it hangs over should be the dead nettle. Yeah, so that should be right. So that should be more than that. Thank you. So let's... so what would I have called that? So now, I'm going to call that 5. That's what I'm going to call that. So, let me just make a note for myself. Yeah, thank you for telling me that. When I first looked at this, I was like, oh my god, this is so chaotic. But actually, it was good. You did well. But I was like,
so bad,
no, it's fine.
Like, oh, no.
It's fine. Listen, that's why I get paid the big bucks for it, Amber. Don't you worry about it.
That's all that matters.
What's that? Question 26. Cool. Thank you. You don't want that. Yeah.
I think I'm a little slow here, but did you mark these ones too? Because was it 15 out of 18? Like, was it you choose 15?
No. However many you do, that's what I graded out of.
So, I don't know, because you marked those ones wrong. Like, you didn't count those ones too.
Yeah.
Okay.
Yeah, you're good. Sorry. I stopped. Yours was the first one I marked, and then at some point I was like, I actually want to take these up, so I'm not gonna. So I just stopped. Sorry about that. Yeah.
How did I get 8.5? Like,
2, 3, 4, 5, 6, 7, 8. This is only 8 options. This is only 8 options. It's not like there's 10 spots, right? And because, do you want my honest answer? Because you should know that's an animal. I think I'm being generous, frankly.
Okay, but can I adjust?
Yes, yeah, of course.
I need to have 3 words.
Okay, but that's a biggie. Listen, I think you got off easy. If you want my honest answer. I'm not trying to be difficult here, but yeah, come on. I think that's fair.
Here it said species.
Yeah, those aren't species. Those are traits, right?
I know.
Those are species.
I know.
Did you?
That's okay.
Did you?
By the way, you did very well. Ruba, I feel like you're so self-critical, or like so stressed out, but you did very well. There you go. It's not so bad. You're so worried about it. It's not so bad.
That's A.
Okay, but that's not so bad.
But it doesn't have a segmented body, right? But like the body.
Yeah, it does. Yeah, worm actually does have a segmented
But
body.
it has legs too, though.
No, it doesn't. Where it's legs?
But
You ever seen a worm with legs?
Yeah.
You've never. No, you
Yeah.
haven't.
It does. Did you ever finish this?
No.
And then
Things are going to be late. Get lowest priority. I will mark it. I promise. Probably tonight. I know. It's okay. I'm not mad. I'm just telling you. Yeah. No, I'm not trying to be difficult. I genuinely think I'm being fair there. mostly on the right track. And I took a few different, like there were a few different configurations here I was happy to take, but I'm not prepared to accept that has legs. Because it doesn't, right? Like there's no legs on there. Yeah, no. Oh, sure. So let me explain. When you do the cladograms, this axis, depending on how you draw them, but if you're doing it this way, this axis really represents time. So like this would imply that like the lamprey existed like back in time before humans, right? But they should all kind of like they're all modern. You can even draw it here and say like you could use this to say like when they came into evolution, but they're still all modern species, so they still all exist at the same time. But other than that, you did great. Yeah, everything lined up well.
I don't get like why up here.
Yeah, so I'll show you. I'll just draw it myself. So like if I'd been drawing this, what I would have done was said. I always go sideways, but I'll try to match yours, just for simplicity's sake here. So how did you do it? Yeah, so if we did, this is like a common ancestor, right? So this is some ancestor that's common to all of them, right? And then if we look up here, we're going to say, okay, that's a modern human, right? Okay, great. So we'll say, on the evolution of the modern human, where did each of these branch off? So we go like that. And I'd say, okay, well, the first thing to branch off is the lamprey, but where you want that to go, you still want it to live up like that high, because these are species that still exist in modern times, right? And then the next one, the tuna, I'd go here and I'd say, okay, and then we have the branching off. But again, we're not showing some like old extinct species, right? Like the tuna still exists today. And then each of the others. Does that kind of make sense?
Oh, yeah,
But that's all. But you did very well. Sorry?
But you took off my.
Yeah. Okay, well, hold up. That's a 90%. So, like, just to be clear, like, oh, give me a sec, because I do want to clear something up, because I don't want you to feel like, so like 50 to 60%, we call like level 1, right? And that is what we say is like not meeting the expectation, right? Not meeting provincial standard, but like showing signs of development, right? So we'd typically call that kind of like developing, right? And then like from 60 to 70, we say that that's like approaching the standard, right? Right? And then from 70 to 80, we say that's meeting the standard. In other words, if you answer the question correctly, that's this, right? Like that's living there, not like, and then everything from there is exceeding the standard, right? Does that make sense? So just keep in mind that like when you're like, oh, you took off a mark, that's predicated on the assumption that like the default is that somebody has 100% and I'm taking away marks when they get something wrong, right? But that's not really how I mark. How I mark is to say like, if you basically answered the question, then like that's living here. But then to get a mark that lives here on any of these kind of open response-ish questions is to make sure that we're like exceeding just the standard. And so that's where things like, is it perfectly neat? Does it have things kind of lined up? And does that make sense? And that doesn't apply to every question, obviously. But in other words, like if you get a 9 out of 10 on something, that's not a criticism. That means that I think you are exceeding the standard. That is complimentary. On the other hand, to get like perfect on something.
I didn't get the proof that.
Let's see. Yeah, so, oh, sorry, other side. Yeah, so, a photographer in sales. He photographed York in its natural habitat and bought the greenhouses for his company in 2003.
But 24 and 3 is, I don't know, I counted, it's not 20 years.
You're going to make the argument because it's 21?
Oh, yeah, sorry.
I mean, listen, I'm that's fair. I can't argue with that. I suppose I should have said more than 20 years ago. Though, to be honest, I wrote that question. I made this test a few years ago and didn't end up using that article. So I used it this year. But that's fair if that's your argument. But, Aruba, you did very well. Okay? You did great. But listen, hundreds are very hard to come by with me. We don't see a lot of them. So don't feel like you're doing poorly when you don't get them. It's just not something I give a lot of out. All right? Probably not going to happen
I
much.
forgot my homework for first period.
Oh, great. It is. It should be up on my lecture, right? On the. Hi. Yes, of course you can.
The note for Friday and the note for Friday. Is it this?
No. Sorry, let me find it for you. Here, I'll find it for you. Sorry, give me a sec. Yeah, what's up? Another broken one?
Another broken one.
Yeah, that's what
And we need to get one. we are now out of the course that are not broken.
No, I just picked up a new pad.
Okay, so because we need to swap out the one it's busted for us. So we have to do that.
Today bio.
We go together, hopefully. It's called cleaning your
As soon as the back of my jaws rang, I just hit forward and then. Thank you. Oh, I have one more question. I do it. You think
Good job, Cassie.
that's
today.
yeah.
That's all?
Homework we kind of.
Oh yeah, I meant to tell you. I
We
found
looked at
my cart.
Yeah.
In the library. One of the kids didn't return it.
I think I can guess.
So can everybody else. I walked in, it was the rest of the class in there. And I walked in, I'm like, are you using this card? She goes, no, I haven't booked cards yet this year. And I said, oh, perfect, I'm taking it. And she goes, I left it here next year.
I did the assignment. I tried the assignment. Okay, is the next unit gonna be better?
Oh, on this one? 97, I think. This one we got 97. Average was 81? No, average was 79. Average was 76, I want to say. 80, 90. No one got 100. I want to say 90. I think somebody in Chem got 98. Is that you? I think you're the highest. So it might have been at 99 and then nobody got 100. I think you're the highest in Chem. Hey, that's pretty good. Aruba, are you competitive?
Oh, Aruba's competitive, yeah.
Good, I like that.
That's good.
Also, do you always match your shoes to your shirt? Because, can I tell you, it's a good ensemble today. Nah, you're doing great, kid. Keep it up.
It's nice to have a little bit of academic competition.
Yeah, I'm not used to the mark rubbers. I'm out of practice with them. That's good. It's good for you.